Hi everyone, this is Manish. I'm a PhD student here at University of Florida in Department of Electrical and Computer Engineering, and I'll be doing a probably one or two more short videos on a class project that I did for a pattern recognition class taught by Dr. Wu, as you can see here on his website. So this is the class website. My project is on muscle skeletal muscle disease perimysium annotation. This is actually a part of my lab work and also something that is related to this class. So let's get started. So yeah, here I am. My lab is BITI2 lab. If you want to check it out, my advisor is Dr. Lin Yang. He is with Department of Biomedical Engineering. This is a brief overview of the presentation that I'll be doing in this short video. I'll be briefly presenting the background of the work that is presented here. I'll present some of the related work that has been done in the past. I'll be overviewing the convolutional neural network based architecture that I have used for this particular work. I'll describe what are the training parameters and how I created the data sets, how many data sets did I use, and some of the parameter tuning that have been done in this work. And finally, the testing approach, which I have undertaken doing a sliding window. I'll briefly talk about it and also a few of the parameter settings that has been done for the testing. And finally, I'll present some evaluation work that I have done, comparative studies, as well as some qualitative results that I have gotten out of this work. I'll just present it. We'll see that the presented work is much better than what has been done in the past. And finally, I will conclude my work and briefly outline a few of the future work that can be extended from this work. So yeah, for the background, the idiopathic inflammatory myopathy is what is known as is a rare form of muscle inflammatory disease, and it basically causes the muscle weakening and pain, and it affects around 5 to 10 million people. I mean, 5 to 10 people per million. I'm sorry, and the average diagnosis is so far has been found to be between 40 to 50, and a male to female ratio of 2.1. And one of the interesting thing is the biology community have recently identified that the myopathies exhibit some sort of clinical manifestation, particularly mononuclear uh, infiltration in the region of perimysium, and the the accurate delineation of the perimysium will help to provide support to characterize this nucleus infiltration in the region of muscle cells, as well as help will be helpful for effective diagnosis and prognosis of the disease. So far, the annotation process in LARS have been in manual or in semi manual mode, which is really time consuming and it's subjective based on the person who is doing the annotation. And the prevailing solution to the problem is using the computer aided algorithms, and this will be more effective when you have to deal with a really large number of digitized muscle specimens. So here is an example of the problem I'm talking about. So this has been taken from a paper published. Here is the URL for that particular paper. So as you can see, this is a cross section area of a normal muscle cell as well as the different types of myopathies. This is the perimysium region. That is the region between different muscle fibers. As you can see, in the normal muscle cells is pretty clear, and the almost all of the muscle cells are in a healthy state. But in these three different types of I'm sorry, there's three different types of myositis, namely polymyositis, dermatomyositis, and the inclusion body myositis. As you can see that there are lots of mononuclear cell infiltration into the region of cells. Some of the cells are like really degenerating with a smaller number of with the cells. I mean the cells, muscle cells being relatively smaller, distributed toward the region of the perimysium. And also there you can see different vacuoles that are invading the muscle cells. So our goal in this particular project is just to identify these perimysium regions so that we can do further analysis on these digitized muscle skeletal muscle images. There have been a few work in this area, not particularly in the field of muscle disease, but in the area of histopathology images, and it's relatively growing area nowadays, developing computer-aided diagnostic system and doing image processing or developing computer-aided algorithms in the digitized histopathological images. They can broadly be divided into two different types. One that basically uses handcrafted features as textures or the color features based on the type of image you are dealing with. Here are a few of the listings that have done a pretty good job in this area. Zhu et al. He recently proposed a multiple instance learning method to do the pixel-wise segmentation. His work also elaborates this to doing a complete annotation, reason-based annotation, as well as the complete diagnosis. And Kong et al. Also proposed a reason of interest annotation methods based on color texture features using the expectation maximization algorithm. Similarly, there have been a growing interest in the area of deep learning or also the learning-based algorithms, particularly in the field of deep learning in recent years. And much of the work has been done in the area of the natural images, where the ImageNet is probably the biggest one that has been done so far in 2012, since when the deep learning has been growing very rapidly. And in the area of medical science or the biomedical engineering or in the histopathological image analysis, Cruz Rao et al. Have demonstrated a use of deep neural network for automated basal cell carcinoma cancer detection. Similarly, Li et al. And has developed a unified deep representation learning model to automate the prostate MR image segmentation. Similarly, recently, Sertian et al. Developed a deep neural network for the detection of mitosis, which is a pretty challenging work. But none of this work particularly deals with skeletal muscle disease, and they are significantly different than any of the other histopathological images. And one of the major challenges, as you can see in this image, is that much of the region in the perimyxium region, more or less, is similar to the other extracellular matrix part of the skeletal muscles, in particular in the mycium region of the muscle cells. So this is pretty challenging work to be done by automated algorithm. So what I propose to do is to use a convolutional neural network that has been that has shown a pretty good work. I mean, pretty good results in both the natural image as well as the in the field of histopathological images. The biggest challenge in doing this implementation in my particular problem is that the the regions are sometimes relatively really narrow. It's really hard to distinguish between the perimyxium region and the endomyxium regions, and to identify like to formulate the problem to do the to the pixel, to do the annotation is pretty hard in itself. So for my formulation, what I did is I just I trained a convolutional neural network with these many layers, which I'll describe briefly later on. Uh, what I basically did is I will also describe this further in my demo part later on, how the negative patch, how I got all the training data and how I got the negative and positive samples. So for now, what I can explain is like, each of these patches that you can see here, all the positive patches that has been explained onto the right side of this architecture, they are basically distinguishing each pixels that are centered to that patch. So each pixel basically is represented by a neighborhood information of around 32 by 32 in my case, and it based on that patch, the convolutional neural network will basically label each pixel as whether being in the perimyxium region or in the non-perimyxium region. So that means if my final goal is to predict each pixel to be in the perimyxium region or the non-perimyxium region. So to train the model accordingly, I cropped. I created a data sets of almost around 200,000 or 300,000 data. For that, what I did is I basically cropped the smaller patches centered on the perimyxium region to be the positive patches, and the patches that are not centered on the perimyxium region, they are the negative patches. So this is the overall architecture of the convolutional neural network that I used. So convolutional neural network, for those of you who don't know about it, basically consists of consecutive layers of convolution layers and the max pooling layers and few of the fully connected layers. This is the very basic layout, uh, because this architecture can go even deeper. That's why it's called deep neural network. And each of the layers that you can see here, they are not predefined. Basically the learning algorithm will train the whole system, and each of these weights belonging to each of these hidden units will be by the learned by the architecture itself. One of the biggest advantages of doing annotation in this way is like, you will not, as there will be a variety of stent digital skeletal muscle images. So if the one of the feature that was one of the handcrafted feature, even if I could identify one, if works on one type of image may not work properly on the other type. So if you train the whole of the feature extraction process as well as the classification process in a single lump like this, the good thing would be that in the whole I don't need to worry about the kind of features that I need to model, and it will easily handle different types of images that I may come up with, or that I may get in the future in the testing phase. Okay, so let's describe this convolutional layer a little bit. So for my implementation, what I did is I just my input is a 32 by 32 patch of three channels, red, green, and blue, basically a raw RGB values of the images that are cropped into a 32 by 32 size, and that is fed into a convolutional layer that is that has a kernel of size 5 by 5 with around 6 feature maps. The stack of feature maps is 6 for the convolution layer, which is basically followed by a max pooling layer. This is max pooling layer has been uh, I mean the biggest growth, I mean biggest introduction in the field of convolutional neural network since the introduction of the um, since the introduction of max pooling layer, the convolutional neural network performance has drastically improved. So what basically this layer does is it simply uh, acts as a feature dimension reduction kind of layer in the convolution layer, and in the I mean the convolutional neural network layer in the overall network, and the convolutional network will simply capture the local image information within a small boundary like this, as shown in the image here. As shown in the image here. So it just captures the local information within that window, and it just creates a multiple feature maps like such. And max pooling is a non-trainable layers which basically summarizes the information within a small window like shown here, to do a further dimension reduction. So we have, for my implementation, I have stacked a two convolution max pooling layer as such. For the first layer has 6 feature maps, and the second layer has 12 feature maps. And finally is followed by a fully connected layer of 64, which is basically linearizes or flattens the features, flattens the output on this particular area so that we can use it for the final analysis to form a single feature vector basically. And it has 64 dimension, and finally we'll have an output layer which is a softmax or the normalized which is basically a softmax layer or the normalized exponential function that gives the probability of each path to be whether positive or negative. So this is my overall architecture. And for the training, what I did is like since all the digitized specimens are scanned by a microscope at different levels of uh, scanning scales. So to deal with that, what I did is I trained my model to have with a multi-scale training data. What that this basically means is that for each central picture, I cropped square patches of size 32 by 32, 28 by 28, 64 by 64 at different scales, and then resize them, all of them, to form a standard size of 32 by 32, and fed all of that data into the convolutional neural network to train. And as I already explained, all those square patches are centered on the pixel in the perimycium region or the perimycium non-perimycium region, forming my positive and negative data sets. My training data that I gathered is around 300,000. It's pretty decent for the architecture as simple as this. And the reason I chose this architecture is to keep the architecture as small as possible and to without compromising the final performance. This will basically help to reduce the computation time. And I ran all of my overall testing and training on the MATLAB code without graphical processing unit. So overall training took me around 8 hours. So you can imagine if I grow the training data size or even grow the architecture, or if I went deeper, then the competition cost would be really high, and it will take a few days to do the training, and even the testing will take longer since my testing has not been optimized so far. For the weight initialization, I just did a uniform random sampling, and then for the training data, I normalized it to be the mean and to be have a unit to have unit mean and standard with the mean and the standard deviation. I normalize my data to be basically within 0 and 1, and this is basically done for the faster convergence. And I have set the learning rate for my network to be 0.1, and this is pretty important parameter in training my convolutional neural network, as this decides how fast my training of the neural network converges, and whether it will overfit or not. So the basic initiation I took is like I decayed the learning rate on each epoch, and also added a early stopping criteria to avoid the overfeeding. So what this basically means is like I run the training on the convolutional network for few epochs and test it on a validation data set. So if I don't see any increase in the validation data set performance, then I will basically stop at that point. And this for each epoch, the batch size I had chose, not for each epoch, but the smaller batch size that I have chosen for each run is of 100. So what basically the training does is runs a feed forward algorithm and then the back propagation for all the whole data set, but it doesn't run it on the batch mode. It just runs on this test. It runs on the online mode. It just runs in a smaller mini batches each time. So until it goes through all the data to form one complete epoch. So the batch size I have chosen is 100, which is pretty decent, which works pretty decent. And even if I increase or decrease the batch size between this a little bit less or more, having this being a median point, it doesn't affect much of the performance. Finally, the momentum, which is another parameter that I need to deal with. I have set it to be constant of 0.5, which doesn't have much effect in my particular training. For the testing, what I did is like I did a sliding window. So basically, for a whole image, what I did is I just put a smaller window of size 32 by 32 on each center on each pixel. And I label that as pixel based on the classification prediction given by my trained model. It's a pixel-wise classification, and I ignore the boundary pixels. Another way of doing it is replicating the boundary pixel, but for my implementation, I basically ignored it. And the testing image has been normalized in the same way as the training data. And the softmax layer is the final output layer for the convolutional neural network that basically gives the probability of the center pixel or that particular patch to be in the peri-matching region or not. And the final prediction label for the patch is done based on the probability, basically threshold the probability map to see whether each pixel is on the peri-matching region or on the non-peri-matching region. So to evaluate my performance, what I did is I did some qualitative as well as quantitative comparisons. I compared my method with two of the following state-of-the-art large-scale support vector machines that has been trained on raw RGB images like I did for the convolutional neural network, and also train the same large-scale SVM on the local binary pattern feature. This basically captures the local texture information within an image, which is pretty popular in the histopathological images because they show the variation in texture pretty widely. So these are some of the qualitative evaluations. So the one on the left hand side, on the extreme left, is the result of the convolutional neural network, while those on the right to R from the large scale SVM. The middle one is based on just on the raw pixel image, and the on toward the right hand side is based on the local binary pattern. As you can see, there are some of the regions that are pretty narrower and are more or less similar to the Indo-Mysian region, as I already explained. They are handled pretty well by the convolutional neural networks, whereas in these other two methods, they are ignored, or they are not so well annotated. Of course, this is just a preliminary results. There is scope for improvement, but still this is a pretty huge improvement. I mean, pretty huge result just for the first time of the evaluation. And to evaluate the performance of my method quantitatively, what I did is I tested the performance matrix, fairly popular performance matrix, precision recall, and F1 score, which are defined as such. Here the NTP is the total number of true positive samples after classification, and this is true positive, false positive, true positive, and false negatives. And the F1 score is defined based on the precision and the recall. Um, I also evaluated the convolutional neural network to map the area of accuracy of, I mean, area under the curve to measure the accuracy of the classification of pixel-wise classification. This is the plot for the ROC plot for the convolutional neural network, and the area under the curve that I report is 0.98, which is pretty decent for the pixel-wise classification accuracy. And as you can see on this particular table, I just listed the mean and the standard deviation of the precision recall and F1 score. The CNN consistently performs better than other two method, one of which is based on just raw RGB pixel, and other one based on the texture feature or the local binary patterns. So this, even the margin is relatively huge. Um, this is pretty encouraging, and this tells how good the deep neural network are, provided that you train it properly, and you have enough training data to train the network, and also depends on how deep you can go with the network. The further deeper you go, the better performance you'll get. But for our particular case, also depends on the size of the window that you are using to do the annotation testing and training. If you use to add it further, so if you use a smaller window, then I'm hoping that I'll be able to even improve the performance in these areas where there seems to be a relatively non-continuous annotation results. But these are pretty hard because even if a manual annotation is to be done on this area, it's relatively really harder for even human annotation to be done. But this result here is pretty good and it's pretty decent, and just based on this, we can do a further analysis that can. I'm pretty sure we can do a pretty decent diagnosis or classification just based on the distribution of the nucleus on these regions. So to finally conclude my work, I have proposed a convolutional neural network based on automatic convolutional neural network based automatic perimysium annotation. And the approach is basically a pixel-wise classification. I have trained my network to handle the multi-scale data, and the experimental result, as I demonstrated, shows the efficacy of the proposed method. And for the future work, what I plan to do is, my testing is by far the sliding window. So I basically am sliding the trained model on each pixel on the large data set. It takes one of these complete annotation roughly takes around 1 or 2 hours. So it's pretty big time to do the testing. So my future work is to find out a relatively faster sliding window method, or some other method that has already been developed there, or maybe find out do this testing on a distributed network somehow. And the other way is the other future work that I plan to do is to train a multiple convolutional neural network instead of just one to handle the multi-scale data, so that I can split the data into different scales and then basically train one convolutional neural network for one scale, and then somehow manage to merge all the information together at the fully connected layer to get the classification accuracy. Um, and I also plan to concentrate on adding some further analysis on this results, so that I can do a subsequent classification of my pattern, which is my ultimate goal. So guys, thank you for listening to my work. I'll be doing one more video to show my overall code architecture and everything. So I will see you guys soon. Thanks.